Hello everyone. I welcome you all to Biology Lessons with Kalyani. Today we are going to start with a new topic, that is lipids, under the chapter Biomolecules, Class 11 Biology. Let's see what's there in the syllabus first. So this is the screenshot of your textbook, and these are the learning outcomes which we are going to touch up today. So what are lipids? Lipids are a broad group of compounds or a heterogeneous group of organic compounds which are fatty in nature or oily or wax-like. Now these compounds are typically not soluble in water or hydrophobic in nature, but are soluble in different organic solvents. What are the organic solvents in which they are soluble? Examples can be ether, chloroform, benzene, right? These lipids are the foundational components of all the living organisms. What does it mean? All the living organisms, they are composed of cells, and all the cells contain membrane, and membrane consists of lipids and proteins. Hence, we can say that lipids are the foundational components of all living organisms. Now, if we talk about the size of the lipids, it is approximately 800 Daltons or less than that. If you remember, in previous sections we read that lipids are found in the acid insoluble fraction or the retentate, even though their size is small. Why? Because they have a tendency to form the aggregates. Talking about the examples of lipids, I already told that they comprise of a broad group of compounds. So examples of lipids can be fats, oils, sterols, phospholipid, glycolipids, and etc. Now, if we have to talk about the functions of the lipid, phospholipid, they act as the integral part of the cell membrane. Lipids can also act as chemical messengers. Example can be all the steroid hormones, thromboxanes, leukotrienes, sphingolipids in nerve conduction, and prostaglandins. Why lipids can act as chemical messengers is because of their hydrophobic nature. They can easily pass through the cell membrane and hence help in the signaling pathway. Third function is storage of energy. Lipids can also store the extra energy in the form of triglycerides. Whenever the body needs it, they can be converted back into the usable energy. Now, this takes place in the process of gluconeogenesis and can happen in the liver, and it can also happen in the adipose tissues. Okay. Now they can be precursors for all the different steroid hormones. Examples can be testosterone, estrogen, cortisol, right? Next is they can help in the absorption of the fat soluble vitamins. What can be the fat soluble vitamins? That can be vitamin A, D, E, K, right? Finally, lipids can also help to insulate the body and protection. Lipids, we know, they make up the adipose tissue or the fat, and the adipose tissue, they help in trapping the air between them because they are hydrophobic in nature, and they are also a very bad conductor of heat. That's how they help in maintaining the body temperature and body insulation. I hope you are clear about the functions of the lipids. Let's move on to the next slide. So till now, we have covered the introduction part of the lipids. Now let us study about the classification of lipids. Traditionally, lipids were classified into simple lipids, compound lipids, derived lipids, and miscellaneous lipids. So what are simple lipids? Simple lipids are esters of fatty acid and alcohol. So what comes under this? All the fats and oils we see and the waxes, they are example of the simple lipid. Next category is compound lipids. What are compound lipids? Compound lipids are also the esters of fatty acid and alcohol, but there is an additional functional group which is attached to them. Those functional groups can be phosphate groups. Then the lipid is called as phospholipid. If the functional group attached is carbohydrate, the type of lipid formed is called as glycolipid. And if a protein group is attached to the lipids, they are called as lipoproteins. Right? These compound lipids can also be called as conjugate lipids. Why they are called as conjugate lipids? Because an additional functional group is attached or conjugated to the lipids. Clear? The third category of lipids is derived lipids. Now what are derived lipids? Derived lipids are those which are produced by the hydrolysis of both simple lipids and compound lipids. For example, if we take fats, then they are made up of fatty acid and alcohol. We hydrolyze them, so we obtain fats and fatty acid and alcohol. Now these fatty acids possess the characteristic of lipids. Hence, they can be categorized under the derived lipids. So examples of derived lipids can be fatty acid, sterols, eicosanoids and ketonic body. In your syllabus, we would be studying more about fatty acids and sterols. The fourth category of lipid is miscellaneous lipids. So what does this comprise of? Now, if the compound does not fall under any such category, simple lipids, compound lipids or derived lipids, but that possesses the characteristics like lipids. For example, it is immiscible in water and it dissolves in organic solvent. Then such substances are put in a miscellaneous lipid category. Examples of them can be squalene and carotenoids. I hope it's clear. Let's move on to next slide. Next is the modern classification of lipids. Also, we can call it as lipid map classification. Under this, lipids can be divided into 8 main types. First is fatty acids. Fatty acid, what does that mean? They will have an acid group and they are fatty. Fatty means they will have a very long hydrocarbon chain. They are typically given by the formula RCOOH, where R is the long alkyl chain or a hydrocarbon chain. Second is glycerolipids. Glycerolipids are nothing but esters of fatty acid and glycerol. Third is glycerophospholipids. Glycerophospholipids means they are the glycerolipids and to them additionally a phosphate group is attached. Fourth category is sphingolipids. Sphingolipids are also ester of fatty acid and alcohol, but here instead of glycerol we use the alcohol sphingosine. These sphingolipids are useful in the nerve conduction. Then is sterol lipids. All the sterols and their derivatives come under this category. Next is saccharolipids. Saccaro, it means the carbohydrate. So fatty acids to which a sugar molecule or carbohydrate is attached are known as saccharolipids. Then prenol lipids. Prenol lipids are nothing but lipids which have an isoprene unit. Final type is polyketides. Polyketides are lipid-like substances which are secreted by some bacteria, fungi and plants. Now, example can be antibiotic, erythromycin. So, that will come under the category polyketides. So, these are all the 8 main types of lipids. We will be reading about fatty acid, glycerolipid and glycerophospholipid and sterol also in detail. I hope you are now clear with the classification of lipids. Let's do fatty acids in detail. Now, fatty acid. Acid means they will have COOH group and fatty means R or the functional group is a long fatty hydrocarbon group, right? So, fatty acids are long fatty hydrocarbon chain or alkyl chain which is attached to a carboxyl group. They can be example of derived lipid because when simple lipids such as fats and oils are hydrolyzed, we obtain fatty acid and alcohol. So, they are example of derived lipid. Now, we can classify fatty acid on the basis of single or double bond present or saturation level as saturated fatty acid and unsaturated fatty acids. What are saturated fatty acids? Saturated fatty acids are those which have single bond present. They don't have any double bond in them. And unsaturated fatty acid, the name suggests they can have, in addition to the single bond, they can have one or more than one double bonds. Now, saturated fatty acids are generally solid at the room temperature. Why? Because they can pack very nicely. That's why they can be solid at the room temperature and melting point of them is also relatively high. The unsaturated fatty acids, in contrast, are liquid at the room temperature. Why? Because their packing efficiency is less. Because the double bond which is present will distort the molecule a little so that they occupy a little bit higher space or the packing efficiency is less in case of the unsaturated fatty acid. So melting point of these are less and they exist as liquid at room temperature. If we talk about the example, ghee is an example of saturated fatty acid. All the fats are example of it and unsaturated fatty acid, their example can be oils. Now, saturated fatty acid are the bad fats because they increase the blood cholesterol and unsaturated fatty acid are the good fats because they decrease the blood cholesterol. Clear? Okay. So, until now, you know what are fatty acids and you also know differences between saturated fatty acid and unsaturated fatty acid. Let's now look at few examples of saturated fatty acid and the formula to write them also. Okay. So, if we have to write the formula for saturated fatty acid, it would be CnH2nO2. So, what is N here? N is the number of carbon atoms. Let's take palmitic acid example first. Palmitic acid has 16 carbon atoms. Put it in the formula C16H2 cross 16, that is 32. O2 is like that we have to write. So the formula becomes C16H32O2. Clear? Now let's try to write it in the form of RCOOH. Okay? First write COH. Now what is remaining? Remaining, it should have 15 carbon atoms. The R group should have 15 carbon atoms because COH, if we include total, should be 16, right? So first write a methyl group and then satisfy the valency. How much you will get? 1 plus 1, 2 is gone. So you will have CH2 taken 14 times. So the formula becomes CH3, CH2 14, COH. It's very important question. If directly it is asked, the number of carbon atoms present in palmitic acid is, you have to write 16. But if it is a little twisted and asked, the number of carbon atoms present in the R group of palmitic acid, then you have to exclude the COH group there. So the answer would be number of carbon atoms present in the functional group is 15C. Clear? So likewise, we will see also stearic acid. It has 18 carbon atom. Formula becomes C18H36O2. And the R group, it would have 17 carbon atom. And how we can write it? First write COH, then write methyl. So how many are remaining? CH2 16. Okay. I hope you are clear with writing the formula for these fatty acid. Okay. Okay, now let's discuss unsaturated fatty acid in a little bit detail. Let's write the formula for unsaturated fatty acid. It is CnH2n minus 2xO2. So what is N here? N is the number of carbon atom. And what is X? X is the number of double bonds. So if we take examples of unsaturated fatty acid, they can be oleic acid, which has 18 carbons but one double bond. Linoleic acid, which again has 18 carbons but two double bonds. And then linolenic acid, which has also 18 carbon atom but three double bond. So 18 carbon atom, one double bond is oleic acid, two double bond is linoleic acid, and three double bond is what? Linolenic acid. Clear? Now let's put in the formula. For oleic acid, it would be C18, H2 cross 18 is 36 minus 2 is 34. So C18, H34O2. That's a formula of oleic acid. If you have to write linoleic acid, it would be C18, H36 minus 2 cross 2, 4. That makes 36 minus 4 is 32. So C18H32O2. That's the formula for linoleic acid. Similarly, write for linolenic acid, it would be C18H2 cross 18 is 36. 36 minus how much? 2 cross 3, that is 6. 36 minus 6 makes 30. So the formula becomes C18H30O2. Clear? Now, one more thing is the molecules which have only one double bond, we call it monounsaturated fatty acid. It has more than one bond, more than one double bond, then we call it polyunsaturated fatty acid. Now, these polyunsaturated fatty acid, they are essential fatty acid. That means the body cannot produce them and you have to outsource it in the form of diet. Okay, let's look into another very important example, that is arachidonic acid. Often question comes from that. Arachidonic acid is an example of unsaturated fatty acid. That is first important point. Second important point is it has 20 carbon. And third is it has 4 double bonds. And that is also an example of PUFA or polyunsaturated fatty acids. Right? So the examples and names of all the saturated and unsaturated fatty acid is very important. Right? I hope this is clear. Okay, I hope now fatty acid is clear for you guys. Let's move on to the next type of lipids, that is glyceride. Under this we are going to cover monoglyceride, diglyceride and triglyceride. Now glycerides are basically examples of simple lipids. I have already told What are simple lipids? They are nothing but esters of fatty acid and alcohol. So what happens here? Let's look at the reaction. Fatty acid is nothing but RCOH and alcohol is nothing but R-OH. So now one water molecule is released, acid will lose the proton and alcohol will be losing its OH group. Both H plus and OH minus combine, water is released and an ester bond is formed. That is RCOOR. Okay, that is your ester bond. Now, in case of glycerides, the alcohol which is taken is glycerol. So, glycerides are essentially the esters of fatty acid and glycerol. What is glycerol? Glycerol is nothing but trihydroxypropane. It is an example of trihydroxy alcohol. This is the structure for glycerol. Now let's come to the structure part. How to write the structure? Now trihydroxypropane. We all know propane. How many carbon atoms it would have? 3 carbon atoms. So first write 3 carbon atoms. 1, 2, 3. Right? Next, it is trihydroxy. So put 3 OH groups. 1 OH here. Trihydroxypropane. So put 3 OH groups in the propane. And then we know carbon has valency of 4. Satisfy the valency. So it becomes CH2. It becomes H. And this one becomes H2. Clear? This is the structure for glycerol. Okay. Let's move on to the next. Now we discussed till now glycerides are the esters of fatty acid and glycerol. Right? Now depending on how many fatty acids react with the glycerol, we can have monoglyceride, diglyceride or triglyceride. Right? In case of monoglyceride, only one glycerol molecule combines with one fatty acid. See, this is glycerol, this is fatty acid. The fatty acid will lose the protons, glycerol will lose the OH minus ions. Both will combine, water is released, monoester bond is formed, and this is called as your monoglyceride. One glycerol, only one fatty acid will react. Now, if 2 fatty acids will react with the glycerol, there will be 2 ester bonds present, right? So the molecule is called as diglyceride. So the diglyceride has 2 ester bonds and monoglyceride has 1 ester bond. Let's look into the structure of triglyceride now. Until now you are clear with monoglyceride and diglyceride. You will be more clear with them while we write the structure of triglyceride. So let's learn how to make triglycerides. Okay? We know that triglycerides are formed by combination of one glycerol molecule, one glycerol and 3 fatty acids, right? So let's write glycerol first. What is glycerol? Glycerol is trihydroxypropane. So first let's write propane. 1, 2, 3. 3 carbon atoms. Trihydroxy. Let's put the hydroxy group. One here, one on this side. I'm putting this on this side so that it becomes a little less clumsy. And another OH group, let's put it here. Okay, let's satisfy the valency. H2, H, H2. So one glycerol molecule will be interacting with 3 fatty acids. Let's put the first fatty acid. We'll put the functional group as R1 here. R1, C double bond O, OH. Let's put another fatty acid here. R2, C double bond O, OH. Okay. Another fatty acid here, R3, C double bond O, OH. Okay. Let's now remove water. Always acid will lose protons and glycerol will lose OH minus. So OH minus, loss of one water molecule. Okay. Here also acid will lose the proton. OH minus will be lost by the glycerol. Same here also. OH will be lost by glycerol. H will be lost by the acid. So that is minus 3 molecules of water. So what do we get? We get the structure C, 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 H2, H, H2. Okay. Now then write O, C, double bond O, R1, O, C, double bond O, R2, O, C, double bond O, R3. So there are one ester bond, second ester bond, 3 ester bonds. So there are 3 ester bonds present. So it is a triester. So if you are asked to write the formula for monoglyceride, just attach one fatty acid. If diglyceride, attach 2 fatty acid. Triglyceride, attach 3 fatty acid. I hope the structure is clear. Let's move on to the next slide. Now let's look at some other examples of simple lipids. Number 1 is wax. Wax is an example of simple lipid which is formed by combination of fatty acid and alcohol. For wax, the fatty acid we use is palmitic acid and the alcohol is myricil alcohol. Other example of waxes are wax D, cerumen or the earwax, canoline. So these are other example of wax. Wax is important. Wax contains palmitic acid and myricil alcohol. Okay. Other examples of simple lipid is cutin, suberin. Both are found in the plants. We are now clear with the simple lipids. Let's now look at what are conjugated lipids or compound lipids. Okay. Now, conjugated lipids also have a fatty acid and an alcohol group, but additionally they have an extra functional group conjugated to it or attached to it. That extra functional group, what can it be? That can be a phosphate group. If it is so, they are known as phospholipids. If the extra functional group is carbohydrate, then we call it as glycolipids. If it is protein, we call it lipoprotein. Clear? Now let's look at phospholipids in detail. Because phospholipids constitute our membrane, right? They form the membrane bilayer. So their structure is almost similar to triglyceride. In triglyceride, what do? We add one glycerol group to 3 fatty acids, right or not? Here, instead of 3 fatty acids, we will add a glycerol molecule to 2 fatty acids and 1 phosphate group. So how do we write the phosphate group? We know phosphate, what is its valency? Its valency is 5. So put a phosphate and then OH, 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 and then you have a double bond O. So that's how we write a phosphate group, right? So now, how do we write the structure of phospholipid? Write just like the triglyceride. How we write it? glycerol molecule, attach two fatty acids, and in the third position attach a phosphate group. Clear? I have marked the phosphate group in blue. So this is how we write the phospholipids. Now these are essentially found in cell membrane, okay, and they constitute a hydrophilic head which is made up of the phosphate group, and the alkyl chain forms a hydrophobic group. Because it contains two groups, both hydrophilic and hydrophobic, so can we call it amphipathic molecule? Right? So phospholipids are an example of amphipathic molecule. Clear? Let's look into another example of phospholipid, that is lecithin. Okay? Lecithin, the structure of lecithin is given in your CBSE book. Here it is a type of phospholipid wherein we attach phospholipid to a choline molecule. Okay? Now what is choline? Choline is a nitrogen containing compound. Lecithin basically acts as a surfactant and it prevents the alveoli from collapsing. Now how can we write the structure of lecithin? It won't be asked, maybe for your class 11th exam you might be asked to draw its structure, but if it is asked for NEET exam, it's very clear that in lecithin, we know that lecithin is an example of phospholipid. So write phospholipid first. What is phospholipid? We know, like it is just like your triglyceride structure, but it will have two fatty acid and one phosphate group. So write one fatty acid, one ester bond, second ester bond, and in the third position you add a phosphate group. Additionally, what do we add in lecithin? We add the choline group. What is choline group? Choline group is nothing but this group. This consists of CH2 and one nitrogen group to which three methyl group are attached. How do you remember it? Phosphate group, you don't need to remember, it is easy. Then you remember we add two CH2 groups, and to it we attach one nitrogen group, and nitrogen, three alkyl groups are, three methyl groups are added, CH3, right? This is how you can remember the structure of lecithin. So lecithin is very important surfactant. It is present in our alveoli. Okay. Another example of conjugated lipids is sphingolipids. Where do we find sphingolipids? We find sphingolipids in nerve cell or myelin sheath. Here it consists of fatty acid and alcohol, but the alcohol here is sphingosine instead of glycerol. Right? You need to remember the name sphingosine. Okay? I hope phospholipids is clear. Let us now do another important type of lipid, that is sterols. Now sterols essentially have a sterol nucleus. What is sterol nucleus? Which is common in all the sterols? It is made up of 4 close rings. 1, 2, 3, 4. Now these rings, out of them 3 are 6-membered ring, and the fourth one is a 5-membered ring. Now different side chains are attached in different sterols. Okay. Example of a sterol is cholesterol. Now we all know that cholesterol is found only in animals. That's why it is called as zoosterol. It is very important because in the membranes, cholesterol acts as a membrane stabilizer. Other important functions of cholesterol are, it is an important precursor of the steroid hormones and also the fat soluble vitamins like vitamin D, and it also helps in the synthesis of bile salts, right? But cholesterol is also harmful. Extra cholesterol is deposited in the blood vessels as plaques and causes atherosclerosis. To make the cholesterol, acetyl coenzyme A is needed. So that's all about sterols. Now only we have one last thing left, that is how to make the structure of the sterol nucleus and how to draw cholesterol. So let's do this now. I told in the cholesterol there would be one sterol nucleus. So let us draw sterol nucleus first. So we will draw 3 6 membered ring. And one 5 membered ring. Okay. Now for cholesterol, we'll attach a double bond here and one OH group here, and then we will add the side chain. So add side chain between the first and the second ring, then third and the fourth, and to the second position of the fifth member ring, you attach a big side chain almost like the 6-membered ring. Let us do it now. 1, 2, 3, 4, 5, 6. Then the fifth position, attach another side chain, and here also attach another side chain. Right? So this is the structure of cholesterol. Easy to remember. Now let us do a quick count of the carbon atoms. For the sterol nucleus, we'll have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 carbon atoms. So 17 carbons are present in the sterol nucleus. If we consider cholesterol, how many extra are present? Side chains, let us count. 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So extra 10, that makes 10 plus 17, 27 carbons. So that's all about sterols. I hope it is clear. I hope you got something out of the video session today. If you like the content, please do like, share and subscribe my channel. Thank you everyone. Have a good day.